Section 1, photosynthesis, the green engine of life. Plants possess a remarkable ability. They can create their own food, a process known as photosynthesis. This process is the foundation of almost all life on Earth. It all starts with sunlight, carbon dioxide, and water. Through a series of intricate steps, plants transform these simple ingredients into glucose. Glucose is a type of sugar that provides energy for the plant to grow and thrive. Photosynthesis occurs within specialized compartments in plant cells called chloroplasts. These chloroplasts contain a green pigment called chlorophyll. Chlorophyll is what gives plants their characteristic green color. More importantly, it's the key to capturing light energy from the sun. This captured energy powers the entire photosynthetic process. The importance of photosynthesis extends far beyond the plant kingdom. Animals, including humans, depend on plants for food and oxygen. When we eat plants or plant-eating animals, we are indirectly consuming the energy that plants originally captured from the sun. Moreover, photosynthesis releases oxygen as a byproduct, which is essential for the respiration of most living organisms. In essence, photosynthesis is the lifeline of our planet. It sustains the delicate balance of the Earth's ecosystems. Understanding this process is crucial to appreciating the interconnectedness of life. By unlocking the secrets of photosynthesis, we gain a deeper understanding of the natural world and our place within it. Section 2. Capturing sunlight, the light-dependent reactions. The first stage of photosynthesis is all about capturing light energy. This stage, aptly named the light-dependent reactions, takes place within the thylakoid membranes of chloroplasts. Imagine these membranes as intricate folds that house the chlorophyll molecules responsible for absorbing sunlight. When sunlight strikes a chlorophyll molecule, it excites an electron, boosting it to a higher energy level. This excited electron is like a tiny packet of energy, ready to be put to work. It embarks on a journey through a chain of electron carriers embedded within the thylakoid membrane. Think of it as a relay race, with the electron being passed from one carrier to the next. Each transfer releases a small amount of energy, which is used to pump protons across the thylakoid membrane. This pumping action creates a difference in proton concentration, forming a gradient across the membrane. Protons, like water flowing downhill, naturally want to move from an area of high concentration to low concentration. This movement of protons is harnessed by an enzyme called ATP synthase, which acts like a turbine. As protons flow through ATP synthase, it spins, generating ATP, the energy currency of cells. Alongside ATP production, the light-dependent reactions also generate NADPH, another energy carrier molecule. NADPH plays a crucial role in the next stage of photosynthesis, the Calvin cycle. In essence, the light-dependent reactions capture light energy and convert it into chemical energy in the form of ATP and NADPH, setting the stage for the synthesis of sugars. Section 3. Building life's building blocks, the Calvin cycle. The Calvin cycle, the second stage of photosynthesis, is where the magic of sugar synthesis happens. This stage takes place in the stroma, the fluid-filled space surrounding the thylakoid membranes within chloroplasts. Unlike the light-dependent reactions, the Calvin cycle doesn't require light directly. Instead, it relies on the ATP and NADPH generated in the previous stage to power its reactions. Think of the Calvin cycle as a complex assembly line, with each step carefully orchestrated. The process begins with carbon dioxide from the atmosphere entering the cycle. This carbon dioxide is fixed onto an organic molecule called RuBP, ribulose 1,5-bisphosphate, in a reaction catalyzed by the enzyme Rubisco. This carbon fixation step is the initial incorporation of inorganic carbon into organic molecules. The resulting 6-carbon molecule is unstable and quickly breaks down into 2 3-carbon molecules. These molecules undergo a series of transformations fueled by the ATP and NADPH from the light-dependent reactions. Through these transformations, the carbon atoms from the initial carbon dioxide molecule are rearranged and reduced, ultimately forming a molecule called glyceraldehyde 3-phosphate, G3P. G3P is a crucial sugar intermediate. Some G3P molecules exit the Calvin cycle to be used by the plant for energy or to build other organic molecules like glucose, starch, and cellulose. These organic molecules form the structural components of the plant's body and provide energy for growth and reproduction. The remaining G3P molecules stay within the cycle to regenerate RuBP, ensuring the cycle can continue and more carbon dioxide can be fixed. In essence, the Calvin cycle utilizes the energy captured from sunlight in the light-dependent reactions to transform carbon dioxide into sugars. These sugars are the building blocks of life, providing energy and structure for plants and ultimately all organisms that depend on them. Section 4. Photosynthesis in action. Factors that affect the process. Photosynthesis is a dynamic process influenced by various environmental factors. Understanding how these factors affect photosynthesis is crucial to appreciating the adaptability of plants and their role in different ecosystems. Light intensity, carbon dioxide concentration, and temperature are some of the key players that can either accelerate or hinder the rate of photosynthesis. Light intensity plays a direct role in the light-dependent reactions. As light intensity increases, the rate of photosynthesis also increases up to a certain point. Imagine shining a brighter light on a plant. It can capture more energy and produce more ATP and NADPH, fueling the Calvin cycle. However, at very high light intensities, the rate of photosynthesis plateaus. This saturation point occurs because the photosynthetic machinery within the chloroplasts reaches its maximum capacity. Carbon dioxide concentration is another limiting factor. As CO2 levels increase, the rate of photosynthesis also increases because more CO2 is available for fixation in the Calvin cycle. However, similar to light intensity, there's a limit to how much CO2 a plant can utilize. Beyond a certain concentration, further increases in CO2 have little effect on the rate of photosynthesis. Temperature influences the activity of enzymes involved in both the light-dependent reactions and the Calvin cycle. Enzymes are biological catalysts that speed up chemical reactions. They have an optimal temperature range where they function most efficiently. At low temperatures, enzyme activity slows down, reducing the rate of photosynthesis. As temperatures rise, enzyme activity increases, but only up to a certain point. Beyond the optimal range, high temperatures can denature enzymes, causing them to lose their shape and function, which disrupts photosynthesis. Understanding these factors and their interplay helps explain why plants thrive in certain environments and struggle in others. It also highlights the delicate balance of conditions required for optimal photosynthetic activity, which ultimately impacts plant growth, productivity, and the entire ecosystem. Section 5. Adapting to the environment. C4 and CAM photosynthesis. Plants are masters of adaptation, evolving different strategies to thrive in diverse environments. In hot, dry climates where water loss is a major concern, some plants have developed specialized pathways to optimize photosynthesis while conserving water. These adaptations, known as C4 and CAM photosynthesis, are variations on the traditional C3 pathway used by most plants. C4 plants, like corn and sugarcane, have evolved a spatial separation of carbon fixation in the Calvin cycle. They possess specialized leaf anatomy with two distinct cell types involved in photosynthesis: mesophyll cells and bundle sheath cells. In C4 plants, carbon dioxide is first fixed into a 4-carbon compound in the mesophyll cells. This compound is then transported to the bundle sheath cells, where it's decarboxylated, releasing CO2 for use in the Calvin cycle. This spatial separation allows C4 plants to maintain a high concentration of CO2 around rubisco in the bundle sheath cells, even when stomata are partially closed to reduce water loss. CAM crassulacean acid metabolism plants, such as cacti and succulents, take a different approach, employing a temporal separation of carbon fixation and the Calvin cycle. These plants open their stomata at night when temperatures are cooler and humidity is higher, minimizing water loss through transpiration. During the night, CAM plants fix carbon dioxide into organic acids, which are stored in vacuoles within their cells. During the day, when stomata are closed to conserve water, these organic acids are decarboxylated, releasing CO2 for use in the Calvin cycle. Both C4 and CAM photosynthesis are adaptations that allow plants to thrive in hot, arid environments where water availability is limited. C4 plants excel in high light, high temperature conditions, while CAM plants are particularly well suited to extremely arid environments with limited water availability. These adaptations highlight the remarkable plasticity of plants and their ability to modify their photosynthetic processes to suit their ecological niche. Section 6. The global impact of photosynthesis, sustaining life on Earth. Photosynthesis is more than just a process occurring within individual plants. It's a fundamental pillar of life on Earth, shaping the very air we breathe and the food we eat. It's the primary source of energy for almost all ecosystems, driving the intricate web of life that connects organisms across the globe. Understanding the global impact of photosynthesis is crucial to appreciating the interconnectedness of our planet and the importance of preserving this vital process. At its core, photosynthesis is a process of energy conversion, capturing light energy from the sun and converting it into chemical energy stored in the bonds of sugars. This energy flows through ecosystems, starting with producers, plants, and moving up through consumers, animals that eat plants or other animals. With each transfer of energy, some is lost as heat, highlighting the importance of a continuous input of energy from the sun to sustain life. Beyond energy flow, photosynthesis plays a crucial role in regulating Earth's atmosphere. It's the primary source of atmospheric oxygen, a byproduct of the light-dependent reactions. The oxygen released by plants is essential for the respiration of most living organisms, including humans. Conversely, photosynthesis removes carbon dioxide from the atmosphere, acting as a natural carbon sink. This process helps regulate Earth's climate by mitigating the greenhouse effect. However, human activities, particularly the burning of fossil fuels, are releasing vast amounts of carbon dioxide into the atmosphere at an unprecedented rate. This excess CO2 is disrupting the delicate balance of Earth's carbon cycle, leading to climate change. Understanding the role of photosynthesis in carbon sequestration is crucial for developing strategies to mitigate climate change and preserve the health of our planet. In essence, photosynthesis is the engine that drives life on Earth, providing energy, regulating the atmosphere, and supporting the intricate web of interactions within ecosystems. Recognizing the global impact of this vital process highlights the interconnectedness of all living organisms and underscores the importance of protecting and preserving our planet's natural systems.